A Carnegie Institution for Science audio press release from May 28, 2015. Quasi-sexual gene transfer and recombination drives genetic diversity of hot spring bacteria. New work from a team including Carnegie's Devaki Baya and Michelle Davison used massive DNA sequencing of bacterial populations that grow in the hot springs in Yellowstone National Park to determine their genetic diversity and explore the underlying evolutionary dynamics. They found an unexpectedly high degree of sharing and exchange of genetic material between the tiny green photosynthetic cyanobacteria Synechococcus, which are abundant in these scalding, inhospitable environments. The team discovered that the pattern of differences in genome organization between various individuals of the same species indicates that the bacteria transfer DNA, including whole genes, back and forth. This swapping or recombination allows gene variations to spread rapidly through a population. Their findings are published by Science. There's a great deal of small-scale genetic diversity in naturally occurring bacterial populations, as opposed to the carefully managed bacterial clones used in laboratory research and clinical work. Bacterial populations in the natural environment represent a dynamic genetic resource that changes over time, but the quantification of this diversity and the exact mechanisms creating its dynamics has remained elusive. Baya explains that biologists have long been interested in determining the evolutionary and ecological forces that drive the population genetics of bacterial communities. The research team, which also included lead author Michael Rosen, as well as Daniel Fisher, both of the Applied Physics Department at Stanford University, set out to investigate this question by combining the power of so-called deep sequencing, which is highly detailed and extensive DNA sequence determination, with powerful statistical analysis. Several possible scenarios were considered. For instance, one theory predicts that bacterial populations are genetically diverse because they adapt to their surrounding conditions on a very small-scale local level, leading to the establishment of distinct subpopulations called ecotypes. Another possibility was that all of the diversity in the bacterial genes is neutral. No particular version of a gene makes an organism more or less fit for its environment. Bacteria reproduce by asexual division, which means that each new generation is stuck with a nearly exact replica of its sole parent's genetic material. Genetic changes can occur through mutation or the transfer of segments of DNA between individual organisms. Using sophisticated statistical analysis of the massive DNA deep sequencing data enabled the team to trace the evolutionary forces that shape these natural Synechococcus populations. They found that neither models of neutral drift nor the concept of microniches of different ecotypes fit the data. Rather, the population occupies a broad niche that includes a range of environmental conditions. Diversity is created by frequent swapping of genetic material between organisms. This apparently happens often enough that the population can be viewed as quasi-sexual, in comparison to organisms like humans, where the process of sexual reproduction, specifically fertilization, combines genes from two parents. In sexual reproduction, new combinations of genes are the rule. Although this isn't generally true for bacterial populations, for these particular hot spring bacteria, new combinations are also the rule rather than the exception. Since DNA moves between individuals, a new generation won't be stuck with just a copy of its parental genes. Because of this level of variation, natural selection acts on the level of individual genes, not the whole genome. Transfers of DNA happen so much that bacteria can have all sorts of different combinations of genes and gene variants. Baya notes that without deep sequencing and careful analysis, we never would have been able to detect and identify the forces at work, and it will be exciting to discover if these insights extend to other microbial communities. Microbial diversity is found everywhere, from deep-sea vents to the human gut or in association with plant roots. Using methods such as single-cell sequencing, proteomics, and microscopy will allow exploration of this invisible and important world with great accuracy and depth. With the independence for nimble pursuit, Carnegie scientists tackle some of the most profound challenges in modern science, fundamentally changing what's possible for us, our planet, and our universe.